Hello everyone. Today we'll talk about Notch signaling as a therapeutic target for malignancies. As Notch is deregulated in many malignancies, multiple strategies are currently developed and tested in preclinical assays and clinical trials. Inhibitory strategies include diverse forms of gamma secretase inhibitors, including the S3 cleavage and thereby activation of all Notch receptors. Inhibitory antibodies against individual Notch receptors and ligands. Specific inhibitory antibodies masking the S2 cleavage site, thereby blocking ADAM protease-mediated cleavage of the receptors. Inhibitory peptides blocking the formation of functional NICD monotranscription complex. JSI was the first class of Notch inhibitors to reach clinical development in oncology. JSI treatment of T-ALL cell lines resulted in rapid clearance of activated Notch1 protein and effective downregulation of Notch1 target genes in leukemia mouse models, increased the survival. JSIs had a synergistic effect with other chemotherapy in human colon adenocarcinoma cell lines. Clinical benefit from JSIs has also been observed. For example, the Aura JSI MPF 0308414 was found to have promising clinical activity in phase 1 dose-finding study in patients with advanced stage solid tumors. In humans, the dose-limiting toxicity of JSIs is secretory diarrhea. The mechanism is that JSIs also block Notch processing in the gastrointestinal crypts. Patients have also been observed to develop a cutaneous rash after treatment with JSIs in several phase 1 clinical trials. JSIs also have the potential to modulate T-cell responses, and this activity has been used therapeutically in experimental models of the graft versus host disease and aplastic anemia. To limit toxicity, most investigators use intermittent JSI administration regimens. In addition, combining JSIs with glucocorticoids or anti-estrogen agents decrease intestinal toxicity in animal models. Inhibitory antibodies for Notch ligands are also being in clinical trials. For example, inhibition of delta-like-4 Notch signaling is a tool for disrupting angiogenesis. The anti-tumor effect was shown to be the result of dysregulated angiogenesis characterized by increased spreading in endothelial tip cells loading to non-functioning vasculature in the tumor. The most common severe, grade 3 to 4, adverse events were fatigue, headache, hypertension, and nausea. As just mentioned, Notch is promising therapeutic target for T-ALL. JSI treatment can lead to rapid clearance of activated Notch1 protein and transcriptional downregulation of Notch1 target genes, induce G1 cell cycle arrest, and finally decrease the size of T-ALL cell lines. Yet the use of JSIs poses challenges. significant intestinal toxicity in patients because of dual inhibition of Notch1 and Notch2, high level of interleukin-6 and interleukin-8 abrogate or significantly reduce the efficacy of the GSI. GSIs may not be useful in patients with Notch1 HD mutation. TAL patients with loss of BDN are resistant to GSI effects. As a potent target for malignancies, it remains to be determined which cancers are typified by active Notch signaling, what specific roles are performed by components of the Notch signaling pathway in a given tumors, what genes or other pathways synergize with Notch, where application of combinational therapy seems sensible. Make a conclusion: the Notch pathway is complex, and the wide range of consequences in different cancer subtypes makes targeting this pathway challenging. There is much remains to be learned about this complex pathway. Given the key role of Notch in cooperating with multiple signaling networks, particularly in stem cells, it's clear that modulation of this system may offer new therapeutic strategies for hematologic diseases. Thank you.